So hello and welcome to another video in the lecture series entitled Advanced Biomaterials. So in our previous videos we had seen about carbon nanoparticles. We had discussed what are carbon nanoparticles and why are they important in different fields of biomedical engineering and also in the therapeutic and healthcare sector. We have seen how carbon nanoparticles can be used as probes for fluorescent imaging. We have also seen how carbon nanoparticles can be used along with other materials for improving their overall physiological or material properties. So we have also seen about the different preparation techniques of carbon nanoparticles and how they can be synthesized using different sources and using different methods. So in today's video we will be mainly focusing on some of the essential characterization techniques of carbon nanoparticles which are essential for determining whether the carbon nanoparticles have been produced and how well are they synthesized, or what are their physiological properties or their mechanical properties or their materialistic nature of the synthesized carbon nanoparticles. So based on the expected features of the carbon nanoparticles, evaluation of their chemical, physical or physico-chemical characterizations need to be performed. Carbon nanoparticles are characterized by different techniques such as spectroscopy, then microscopy, and other analytical techniques. So these techniques are crucial in terms of defining different types of properties of the carbon nanoparticles. Namely, the techniques include scanning electron microscopy, SEM, atomic force microscopy or AFM. Then we have these are mainly used for studying the defined morphology of the carbon nanoparticles. And also the micro and nanostructure can be determined as well using these techniques. So other techniques such as the dynamic light scattering, so the DLS or the dynamic light scattering techniques, and transmission electron microscopy TEM can be used to determine the hydrous and anhydrous diameter. So hydrous and anhydrous diameter of carbon nanoparticles. Now, this transmission electron microscopy or TEM is also applied for studying the orderedness of the composition on the different patterns of the surface properties or the overall properties of the synthesized carbon nanoparticles. So apart from this transmission electron microscopy, there is another technique called as XRD or X-ray diffraction crystallography. So this XRD is also an important tool for studying the crystallinity of CNPs. So this XRD gives an estimation or determination of the crystallinity or the crystalline properties of the carbon nanoparticles. Now, if you look at the XRD data, there are different peaks which are obtained at different angles. So, it has been reported that a broader peak at 23 degrees and another peak at 41 degree indicated the presence of amorphous carbon and graphitic structure respectively. So the peak at 23 degrees implies or signifies the presence of amorphous carbon, whereas the peak at 41 degrees signifies the presence of graphitic structure in an X-ray diffraction crystallographic data. Now there is another characterization technique or tool which is known as Raman spectroscopy. Which gives an estimation about the specific chemistry of the carbon nanoparticles. So Raman spectroscopy for the carbon nanoparticles synthesized from, as reported in a particular literature, that was carbon soot. So carbon soot was described by the presence of graphitic or G band and D or the diamond band. At 1592 and 1360 centimeter inverse. So centimeter inverse represents the wave numbers. So the presence of graphitic and diamond band present at approximately 1592 and 1360 centimeter inverse. Now, so if the diagram is like intensity in the y-axis and Raman shift in centimeter inverse or the wave number, so we can get a spectra like somewhat like this. So this and this is the D band and the G band. So based on this, the intensity of the diamond band and the intensity by the ratio of the intensity between the diamond and the graphite band equal to 2 gives an indication about the non-crystallinity of CNPs. So these CNPs are non-crystalline in nature, and it also can provide information about the sp3 versus the sp2 hybridization of carbon. Another technique known as the FTIR or Fourier transform infrared spectroscopy is crucial to decipher the functional groups. So this technique is used to decipher the functional groups based on specific stretching vibration of the type of chemical bond which is present in a molecule. So every molecule will have different chemical bonds associated with it, and based on the stretching vibrations of these different chemical bonds, the FTIR or Fourier transform infrared spectroscopy can give an estimation about the different types of bonds which are present in a molecule or are present in a structure. So it has been reported that carbon nanoparticles which have been synthesized from a given source such as agave nectar, a natural carbohydrate source based on this hydrothermal method of synthesis, contains this OH, CH, C double bond O and OCO at wavelengths 3,4 wave number, 2900, 1700 and 1200 centimeter inverse respectively. So these different functional groups, or these different groups, or the stretching of these bonds at these particular wave numbers can be deduced or inferred from the FTIR data. Now there are two other simple yet very convenient or very powerful characterization techniques for synthesis of carbon nanoparticles. So those are UV visible spectroscopy and fluorescence spectroscopy. So the optical properties of these carbon nanoparticles are mainly analyzed through their absorption and emission spectrum obtained from UV visible spectroscopy. So UV visible and fluorescence spectroscopy. So these carbon nanoparticles synthesized from, as reported in the literature, from food caramels such as sucrose, honey, and other caramel based sources show two peaks. So if this is a UV visible spectrum, this is the wavelength in nanometers, and this is absorbance in arbitrary units. So there is the presence of two peaks. Okay. Now this peak, the presence of first peak at about 280 to 300 nanometers correspond to a pi to pi star electronic transition. Now we know that absorbance does happen because of electronic transition. So when a chemical molecule is hit with a UV or a visible ray of an electromagnetic radiation in the UV visible region, so the electrons absorb a part of the energy associated with the beam and get transferred from a lower energy level to a higher energy level. So this transition occurs from a lower energy level to a higher energy level, and this transition is associated with an energy which is absorbed during this process. So from the incident radiation or the incident beam of light, a part of the energy is absorbed to enable these electrons to undergo transition from this lower to higher energy level, and the rest of the energy is emitted. So based on that, this absorbance of these particular electrons of these particular chemical species, the first peak at 280 to 300 nanometers is seen, which corresponds to furfurai, which is a not very specific product for the synthesis of carbon nanoparticles. But there is a second peak which is obtained at about 340 to 380 nanometers. So 340 to 380 nanometers, which corresponds to N to pi star transition. Now this peak at N to pi star which can be attributed to the N to pi star transition is actually significant for the synthesis of carbon nanoparticles. Now the presence of these peaks or the pattern of UV visible absorption changes depending on the types of modifications like polarity of the synthesized carbon nanoparticles, surface passivation, and also the precursor material for the synthesis of carbon nanoparticles. So often it is found that these two peaks are not very nicely resolved. So this spectrum can also appear like one peak and one shoulder. So this absorbance is actually superimposed with the major peak at the lower wavelength, and the N2 pi star is obtained as a slope or as a shoulder. But nonetheless, this will imply the synthesis of carbon nanoparticles. Now, there is another technique, the fluorescence spectroscopy. So, in fluorescence spectroscopy, the rationale behind is that since carbon nanoparticles show the property of autofluorescence, upon exciting the carbon nanoparticles with a wavelength or with a light at a lower wavelength, they will emit light at higher wavelength. Right? So, measurement of photoluminescence is another method for interpreting the optical properties of the carbon nanoparticles. So, this is determined by exciting the carbon nanoparticles at different wavelengths, causing emission at various higher wavelengths. So, excitation at lower wavelengths causes emission at higher wavelengths. Now it has been seen that upon exciting carbon nanoparticles synthesized from agave nectar as carbon source, the excitation wavelength at 365 nanometers gave an emission at 488 nanometers. So this emission pattern can also be a characteristic measure for the synthesis of carbon nanoparticles. So in this video we had actually seen the different methods of carbon nanoparticle synthesis. Now some of these methods are widely or routinely used for measuring the synthesis of measuring the efficacy of carbon nanoparticle synthesis. Now based on the particular type of applications or the way in which we are trying to fabricate the carbon nanoparticles for a specific use or a specific role, we can perform either some of the very specific characterization techniques or a combination of some of these characterization techniques. So some of them are preliminary, while some of them, such as the AFM or TEM, are more more specific to for determining very specific characteristics of the carbon nanoparticles. So with this, we'll end today's video. See you in the next one. Thank you.